Chris, thank you very much, and I really do appreciate the invitation. And I've been impressed for so many years of the progress that's gone on under FNIH and its diverse programs, including in the biomarker space. So this is obviously one session that I think is indicative of the kind of progress that this organization can make. What I'm going to do is, in the next 15 minutes, really just at a pretty high level, because I'm not a biomarker expert. I'll leave that to Chris and to colleagues here. But I'm going to talk a little bit about some of the regulatory challenges and limitations around biomarkers, which I think we have to be cognizant of as we try to make progress in the space, which is clearly so important. But what I'll actually do is spend just 2 minutes talking about some of the broader challenges in the changing landscape in drug development, because I think it provides some context for thinking about the advances we need to make in the biomarker space in neuroscience. Then I'll talk briefly about an overview of biomarkers, and then talk at the end about some of the challenges I think we face. So this is, and I'm sorry for this being a busy slide, but there are so many changes that are going on in the space of drug development and in the regulatory landscape as well. Science is changing, and again, I a bit carrying coals to Newcastle to talk about these things to this group, which you're so well aware of and involved with. But we're clearly seeing just an explosion in our understanding of genetics of disease, the genomic characterization in many diseases, which has led to molecular subtyping across many disease categories, new molecular drivers, which of course has been important in cancer, but we're also seeing this important in genetic subtypes of disease, such as cystic fibrosis. And importantly, new platforms for drug therapies, ASOs, siRNAs, cellular regenerative therapies, very new complex biologics that have allowed druggable targets that were previously undruggable to be druggable. The first siRNA approval, for example, for a drug that included a target in neuropathy for an inherited amyloid disease. We're seeing the types of targets for disease changing, which just reflects that. So many more diseases that are rare, that are finding new therapies, disease subtypes that are being targeted cancer therapies, of course, an explosion in that area. And obviously access has been a key issue, so the rise in biosimilars and, of course, the continuing growth in generic compounds, now moving more to complex generics. In terms of the development and regulatory landscape, we're seeing an increased expectation of the role of patients, which I think has been very important. It's actually led to even further push towards the development of new COAs, looking at a variety of different aspects that are important to patients. An increased focus on biomarkers, and Chris is going to talk about the efforts within FDA on enhancing our BQP, new approval pathways, master protocols, which is something that we're encouraging, which I think in the neuroscience space could be so important, particularly for diseases that are less common and where there really are innovative therapies that need to be examined. Decentralized trials and mobile technologies, an explosion in the growth of mobile technologies with everyone wearing an iWatch, which I don't have, but with an iWatch that could look at GPS locations, accelerometers, vital sign monitoring. How do we incorporate that, which of course would be very impactful for many diseases that are neuromuscular or neurologic in focus, that may enhance our ability to use that as a surrogate endpoint of therapies. How do we do that? How do we incorporate that into new COAs? And I think also the growth in real-world evidence and what kind of endpoints can we derive from real-world evidence and how can we use it. That's something that we're really being asked to consider, and we're really working on with multiple subcommittees at FDA. Now, I just put this together just to say that there really has been a change in how and what drugs have been targeted over the past decade. What I did is I just took the approvals back 10 years ago and then the approvals last year, just to say that, and to a certain extent stating what I think is probably obvious to many people, a dramatic rise in targeted therapies and drugs for rare disease, and a reduction, as you can see, in the non-rare chronic diseases, going from 60 approvals down to 60% approvals down to 30% of the approvals. And what I think is also important is among those 30%, it tends to be more later stage diseases, add-on therapies. So it's not the same types of therapies even in the common chronic disease targeted drugs that we saw 10 years ago. So a big change in the types of drugs that we're seeing, which we have to adapt to in how we think about regulating these drugs. With that introduction, let me just sort of set the stage around biomarkers. And again, I apologize, many of you I think are quite familiar with these definitions, But this is, I think, just a category as to how we think about it. A clinical outcome is what we're looking for registering a drug. So it's something that describes how a patient feels, functions, or survives. So that concept is very important to us. Those are the regulatory endpoints that we're looking for drug approval. Does it improve how a patient feels, functions, or survives? Or are there events that impact how a patient feels, functions, or survives? Those are the regulatory requirements around what's necessary to approve a drug. A clinical outcome assessment assesses, describes, or measures, or reflects a clinical outcome. It could be reported by a clinician, reported by a patient, or by a trained observer. And of course, this is where the new approach in incorporating mobile technologies will come in as we try to figure out how do we incorporate that into an endpoint that can be a COA that we can accept. Now, of course, a biomarker is different. It's a characteristic. It could be reflecting a biologic process, a pathogenic process, a response to drug or to other exposures, and it can be molecular, histologic, radiographic, or physiologic, but it's not a direct assessment of how a patient feels or functions. And I talk more about surrogate endpoints, which of course are so important because potentially they can be used for registering drugs, but again, the hurdle for that is high, and I'll talk about some of the limitations. So a surrogate is a biomarker that predicts, again, it doesn't directly measure, but it predicts a clinical outcome. And so this can potentially be, once accepted, used for the registration of drugs. Now, the framework for this comes out of BEST. This was a group created by the NIH and FDA some years ago, the Biomarker Working Group, that created this categorization because, of course, the definitions of biomarkers were all over the place, of types of biomarkers, and people talked about them in different contexts, in different ways, with different dictionaries, as it were. And so this was an effort to try to come to some standardization of what we mean by different categories of biomarkers. And so this is the categorization. And I've just divided this to say some of these reflect the disease status, the disease process, and some of these are measuring the response to treatment. So the susceptibility or risk biomarker tells us as a patient whether that patient might be at risk for the development of a particular disease. A diagnostic biomarker classifies a patient as having a disease or a subtype of disease. A prognostic biomarker tells us whether patients are at risk of having an event, an outcome of that disease. A monitoring biomarker might look for safety events that might occur with that disease. A predictive biomarker tells us whether that patient might respond to a particular drug therapy. The pharmacodynamic or response biomarker looks at whether the drug is having a particular effect in that disease. And obviously a safety biomarker is looking at the drug effect with regard to safety endpoints for the treatment in that particular patient. Now, I also looked at it in a different way, which is that biomarkers can be across a cascade of different, in the pathogenesis of a disease. They can reflect a causal factor, so for example, HIV titers or an AFB smear. They can be in the immediate pathway of disease, so LDL cholesterol or blood pressure or A1C might be examples of that. They can reflect organ injury, so they might be biomarkers of tissue injury or biomarkers of organ function. And finally, those that we're looking at for more meaningful endpoints that might be used for registration are those that reflect functional measures or clinical function and can obviously lead to important clinical outcomes for us. But each of these has limitations. So, I mean, in terms of looking at causal factors, I can give the example of AFB smear in atypicals. We've been dealing with this recently with an atypical mycobacterial infection where cultures are positive but occur in patients with severe underlying lung disease. And so is that a commensurable, is an infection? Is clearance of that really reflective of improvement in disease, or is it just the microbiologic therapy that's reducing AFB but not changing the course of disease? So how does that really relate to the disease and its outcome? In the causal pathway of disease, we can look at something like LDL cholesterol, which of course is a long accepted biomarker. But on the other hand, do we know that all different therapies that modulate LDL have the same impact on outcome? They may or may not. Genetically, they don't necessarily. And so we have to think about whether LDL is a broad biomarker or could it be more narrow. Reflecting organ injury, this can be relatively nonspecific. So does it reflect the injury from a particular pathogenic process or from a different process or background disease or other influences? So each of these has limitations in how we can translate and understand the interpretation of changes in these biomarkers. Now, I want to just introduce the concept of context of use, which I hope is a concept that's very well understood already by this group. But it reflects the disease setting and in particular how the biomarker is to be used. In particular, which of the categories that I've outlined is the intent of the biomarker use. Individual biomarkers can be used for a wide range of potential purposes. And in fact, over time, those purposes can evolve. And that as data collects and as we understand the biomarker, it might be initially used as a diagnostic biomarker as we understand what the cut point for the definition of a disease might be. Or a prognostic biomarker, does it tell us the likelihood of complications or of outcome events for that particular disease? Or a predictive biomarker, does it explain or predict whether a particular drug will have a response in a particular patient? Or can it be a surrogate endpoint, one that predicts the benefit that the drug will have on a meaningful clinical outcome? And as I said, the uses may evolve over time. One of the things that we do face, and I'll come back to this later, is a desire often for those developing biomarkers to have all of these varying purposes. So sort of 5 or 6 different proposed contexts of uses. But it's often best to start more in a focused way so we can understand the biomarker, and the evolution of data will help define additional purposes over time. So what I want to then turn to is talking a little bit about the limitations that we have with surrogate endpoints. So again, this is not a direct measure of how a patient feels, functions, or survives, but it's intended to reflect and predict clinical benefit, not measure the outcome, but predict the outcome. And so with a surrogate endpoint, when we are dealing with that, we have to make some assumptions. When we're thinking about registering a drug, can we approve this drug? We have to think about the benefit-risk balance, and we have to make some assumptions about how much benefit there is relative to the risk that the drug might pose. And there are challenges of translating from this surrogate measure of predicting benefit to the extent of clinical benefit that a particular patient will obtain. Often in clinical trials that don't have a clinical outcome measure but are using a surrogate endpoint, we have a smaller safety database, so the quantum of information we have about risk may be lower. However, I will say that we still can estimate benefit and risk. We can still try to understand the translation of the surrogate endpoint to benefit and understand risk from the information that we have. And we have to make benefit and risk decisions. I will say sometimes my colleagues at FDA will say, well, it's a surrogate endpoint, so we really don't know what the benefit is. But what I would say that is, in fact, if we are making a benefit-risk conclusion, by definition, we have translated whatever evidence we have from that surrogate endpoint to a quantum of clinical benefit the patient has and have to understand the risk. And we've made a determination that benefit exceeds risk if we've approved the drug. So one of the concerns, of course, always is that biomarkers may fail to predict clinical benefit, and there is always a residual risk that the strength or the presence of the relationship to the clinical endpoint turns out to not have the strength that we thought it would have, or not be valid. And there's certainly many examples of sort of sure thing biomarkers that turn out to be not such sure things. So what are some of the challenges of biomarker development? Well, certainly program-related, I've alluded to already. One of the problems we see is whether there's clarity on the purpose of the biomarker. Many biomarker programs come to us with a whole profusion of contexts of uses that are proposed rather than a more narrow or focused proposal. One of the other issues is the efforts early on analytic validation. Can you rely on the results from this biomarker? Very often it's hard to invest early on trying to make sure that the analytic methodologies are well described and well tuned. But it's critically important for us to be able to ultimately interpret that biomarker. So early analytic validation as an investment is very important. And I would also say lack of cohesive planning. Often the purpose of the program, as I started by saying earlier, and its focus isn't entirely clear. One of the challenges, of course, we see more broadly is the limited understanding of disease pathogenesis. There are often many, many different pathways that may contribute to a particular disease. So which is the most important? What does the drug modulate? Does it modulate a pathway relevant to the course of the disease? These are often very difficult questions to answer, and very often we can go after a particular pathway that seems to be important in the pathogenesis, modulate it, show that the biomarker changes, but ultimately find out that the disease does not. What are the disease characteristics that we have to think about that particularly challenge biomarker development? Well, without doubt, slowly progressive diseases impede biomarker validation. A long course to outcome, how do we then look at early changes and try to predict whether that will have meaningful ultimate benefit on how patients feel, function, or survive? Rare diseases and uncommon diseases are, of course, a challenge. Few patients, often widely distributed in different centers. How do we bring them together so that we can accumulate the evidence that we need? We're dealing more and more with diseases that are that we're recognizing as genetically, genomically diverse, heterogeneous, with multiple subtypes. Well, do the biomarkers predict the benefit or predict whatever we think they're predicting comparably across those subtypes, across those variants? That's something we have to understand. If there's not a clear gold standard for the diagnosis of a disease, then validating a particular biomarker to an outcome will be in the background of a lot of noise of patients who may or may not have that particular disease. And obviously, when we're dealing with remote organ involvement, our ability to do tissue sampling to correlate with biomarkers is quite limited. Now, I do just want to remind you that we think about these in two senses. I've talked about what a biomarker is. It's something that is going to predict a clinical outcome, but isn't a direct measure of a clinical outcome, reflecting how a patient feels, functions, or survives. We consider a validated surrogate endpoint one that we can rely on to approve a drug, but a reasonably likely surrogate where the strength of evidence is more limited is one where we can use it for accelerated approval, but that assumes that there is a definitive study that can be done to show that the drug has the benefit on a clinical outcome that we expect it to have based upon that reasonably likely surrogate endpoint. Now, somewhat of a complex picture, but again, just to bring out the theme that I've talked about already, which is understanding the placement of the biomarker in the cascade from the disease to the outcomes that we're looking at. The biomarker can be along the pathogenic pathway, and so the drug modulates the disease, changes the biomarker, it influences the clinical outcome, an optimal situation, and one where the biomarker may be a very good surrogate endpoint predicting benefit. But the clinical outcome may also lead to changes in biomarker, but so may many other processes change the clinical outcome and modulate those biomarkers. So maybe the relationship to drug therapy is a little bit less firm. Or the biomarker may not be on the same pathway. It may reflect the disease. The drug may modulate it, but it may not relate to the clinical outcome. And so the drug relationship and the changes in the biomarker may not relate as closely to the clinical outcome. Another scenario that we worry about is whether the drug may indeed modulate the biomarker, improve the clinical outcome, but does it represent the full picture of the benefit the patient has? Might there be toxicities that are not seen based upon the effect that the drug has that are not measured in the clinical outcome? So the relationship between disease to biomarker to clinical outcome is a complex one, and as I said before, we really have to think about it as we look at the different categories or placement of biomarkers. Where is this biomarker? What might go wrong as we try to relate the biomarker to the important clinical outcome? So to finish, I just want to mention a few other elements that we have to think about when we consider the challenges of biomarker development. There are clearly many diseases, many neurologic diseases, of course, with unmet needs and with markedly insufficient drug development tools. We need biomarkers to be able to make progress in identifying potential drugs early, identifying the dose of drugs, identifying drugs that are effective and therefore should be approved. We recognize that biomarker development is a long and very resource-intensive process. Hence, the collaboration of organizations like FNIH and others are critical to be able to leverage the resources of multiple different organizations and institutions. It is, of course, a very long and, as I said, resource-intensive process. There's many stakeholders in the mix, And one of the concerns in, I think, many areas of biomarker development is that there are competing interests. There may be commercial interests. There may be different academic sites with different interests. There may be other groups that are involved with different interests, even different stakeholder groups with different interests. Trying to integrate across these is a critical effort so that the resources are not divided. In some areas, I see multiple biomarker programs ongoing, but not necessarily working collaboratively, competing for relatively limited patient resources and financial resources, which clearly will limit the ability to develop biomarkers in an effective and early way. So the infrastructure to align interests into cohesive development programs is absolutely critical, and that's why meetings like this, I think, are so important. So the challenge, and I'll ask you to think about this over the next days, is how do we prioritize biomarker needs, not try to get biomarkers for everything for a particular disease, but what is the most important needs? What's the strategy for biomarker development that we could all sign on to? How do we focus the resources across multiple organizations to get to that strategy implementation and integrate efforts across competing stakeholders who have to work together if we're going to get biomarkers that help us develop drugs for patients who need them so desperately? So I'll stop on that point. Thank you for your attention, and turn it back to Chris, who's going to tell you more about the BQP at FDA. Again, thank you for your attention.